Vanakkam, good morning. In this video, we are going to discuss about the transport across the cell membrane. Transport across the cell membrane may be asked as the long question in the university examination from the general physiology. This is an important topic for the understand the systemic physiology. Active transport is the one type of transport across the cell membrane. The active transport again divided into the two types, primary active transport and secondary active transport. Active transport, it needs energy, mainly ATP. It needs the energy. This also carries the carrier protein and it transport against the concentration or electrical or pressure gradient. It is the uphill movements. So it is transport against the concentration, mainly concentration gradient. Active transport has two types. One is the primary active transport and secondary active transport. Secondary active transport may be co-transport or symport or counter transport and depot. We will see in detail in next slides. Primary active transport, it directly utilizes the energy. So main example for the primary active transport is the sodium potassium ATPase pump. It is present in all the animal cell membrane, human cell membrane. So this is the picture of the sodium potassium pump. It has two subunits, alpha and beta subunits. Alpha subunit has a larger than beta subunit. It has a sodium binding site. Number 1 is the sodium binding site. Number 4 is the phosphorylation site. Number 5 is the ATP binding site. Number 2 is the potassium binding site. Number 3 is the O-bind binding site. So sodium potassium pumps the 3 sodium from the cell to the ECF, intracellular part to the extracellular part, 2 potassium from the extracellular part to the intracellular part. It is important for maintaining the cell volume and cell shape. So the sodium potassium pump catalyzes the ATP dependent transport sodium out of the cell in the exchange for the potassium entry into the cell. Sodium potassium pump the 3 sodium outside and pump 2 potassium inside. So it creates a more negativity inside. It is also play the less important role in the resting membrane potential. So resting membrane potential is always negative. So it creates more negativity inside. Functions of sodium potassium pump: maintain the cell volume. It is important for the maintain the shape and volume of the cell, and it is also important reason for the genesis for the action potential. So it is the electrogenic pump, and it is also store the energy for the secondary active transport. These are the functions of the sodium potassium pump. Sodium potassium pump is a separate 5-mark question. So you go to write the structure and the function and how you transport the sodium outside, how you transport the potassium inside with the diagram. The secondary active transport is the type of active transport. The primary active transport energy is directly utilized, but in the secondary active transport energy is indirectly used. So here the sodium potassium pump the 3 sodium outside, 2 potassium inside. Within the cell it has a less amount of the sodium, so it produces the concentration gradient in the extracellular area or that luminal area. It has a more amount of the sodium compared to the intracellular part. Okay, so this primary active, the sodium potassium pump indirectly helpful to transport this. These are the counter transport. Potassium and hydrogen transported in the opposite direction. Calcium and sodium transport occur in the opposite direction. So this is called as the counter transport or antipode. Here all are transported in the same direction. Sodium and amino acids, sodium and chloride, sodium potassium chloride. These are called as the co-transport or sympode. So secondary active transport may be co-transport or sympode or counter transport or antipode, but it indirectly utilizes the energy. The sodium potassium pump the 3 sodium outside, 2 potassium inside. So inside the cell sodium level is less. Here the sodium level is more. So due to the concentration gradient, transporter like counter transport or co-transport with indirectly use the ATP, sodium potassium pump ATP. So it may be two types, co-transport or sympode. These are the co-transport or sympodes. Same direction they are transporting. Here counter transport or antipodes. The sympodes. Example is the oral regurgitation therapy for the diarrhea treatment, for the rapid absorption and restoration of the body fluid volume. So sodium is reabsorbed along with the glucose, that is a sodium dependent glucose transporters. It uses the energy indirectly. This is the example for the secondary active transport. And sodium potassium chloride sympode occur in the loop of Henle in the kidney nephron. This is also example for the secondary active transport sympode type. Antipode or counter transports carries exchange one solute for the another across the cell membrane. The examples are sodium calcium in the cell membrane, exchange the sodium and calcium, like that sodium and hydrogen in the renal tubule, and anion exchanger, chloride bicarbonate exchanger is present in the RBC cell membrane. These are the examples for the counter transport or antipode. Use the indirectly utilize the energy, the secondary active transports. Antipodes, it is also used in the therapeutic uses for the treatment of the heart failure. Mainly the digitalis, it act on the cardiac muscle, block the sodium potassium ATPase pumps. It decreases the sodium calcium antipodes. It increases the intracellular calcium level. It produces the increase the cardiac muscle contractility, that is called as the positive inotropic effect, mainly used in the heart failure. Passive transports. This is another type of the transport across the cell membrane. The passive transport, no expenditure of the energy molecule, no need of the ATP here, and it takes place along the concentration and pressure gradient. It is high concentration to low concentration or high pressure to the low pressure, and it carrier may or may not be required. Some places carrier needed, some places carrier not needed. The rate is proportional to the concentration difference. The rate of transport is due to the how much concentration is present. Simple diffusion is the one type of passive transport. It is movement of molecules from the higher concentration to the lower concentration till the equilibrium is reached. So here higher concentration, here lower concentration. So it is more amount is transported from the higher concentration to the lower concentration. Diffusion. Lipid soluble substances like oxygen, carbon dioxide from the higher concentration to lower concentration easily diffusible, and alcohol steroid also easily diffusible through the bilayer lipid membrane. Water and electrolytes are little difficult to do the simple diffusion. It is transported through the selective channels. Factors affecting the rate of diffusion: lipid solubility easily diffused, the molecular size and weight, and the temperature. The higher temperature can easily diffused, and thickness of the membrane. The thickness of the membrane is less means easily transported, and a surface area, larger surface area, it transport the more amount by the simple diffusion, and concentration gradient higher to lower, and pressure gradient high pressure to low pressure, and electrical gradient negative to positive side or positive to negative side. So these are the factors affecting the rate of diffusion. The fixed law of diffusion: the rate of diffusion is net rate of diffusion is directly proportional to the concentration gradient of the substance and permeability of the membrane to the substance and surface area of the membrane, and indirectly proportional to the molecular weight of the substance and thickness of the cell membrane. Facilitated diffusion, it is the type of passive transport. It is important for the larger water soluble molecules. It transport along the concentration gradient, but it needs the carrier protein. It is the carrier mediated transport. The receptor site is present in the one side of the cell membrane. The example for the facilitated diffusion is the transport of the glucose through the glucose transporter. This graph depicts the difference between the simple diffusion and facilitated diffusion. The blue line is the simple diffusion. The rate of diffusion is directly proportional to the concentration of the substance. This is a direct. But in the facilitated diffusion is depicted in the red color line. It has the maximum transport, that is called as a Vmax, is present in the facilitated diffusion. Once the transporter is utilized, means the transport is maximum occur at the one place. So this is the difference between the facilitated diffusion and simple diffusion. Features of carrier transport protein. Features of facilitated diffusion. It has a specificity. So transporters use a specific substance. It has a competitive or non-competitive diffusion, because the glucose transporter can block by the fluorescein. It has a saturation. Saturation means transport is occur depend on the availability of the transporters. So receptor can be blocked by the another substance, blocking of the receptors. It has a Vmax, maximum transportation, depend on the transporters. The example for the facilitated diffusions are transport of glucose through the glucose transporters, and amino acid transporters, and galactose transporters. Osmosis is the type of passive transport when the two solutions of different concentration, lower concentration and higher concentrations, are separated by the semi-permeable membrane. It is impermeable to solute and permeable to water. The water molecules diffuse from the solutions having lesser concentration to the higher concentration. That is called as the osmosis. Here, when pressure is applied over this, it prevents the entry of the water molecule in the particular level. That is called as the osmotic pressure. This osmotic pressure is very, very important. So this is maintain the tonicity of the solution. The osmotic pressure is the minimum pressure applied on the solution with higher concentration, which prevent the osmosis. That is osmotic pressure prevent the osmosis depend on the total number of the particles of dissolved solute rather than the type of particles. So the number of particles it maintains the osmotic pressure. The osmoles are expresses the concentration of the osmotically active particles. Okay. 1 osmol means total number of the particles in the 1 gram of the molecules. Okay. So it is the weight of non-diffusible substance per kg of the water. Applied aspects of the osmolality. The isotonic, hypotonic solutions. The tonicity is isotonic means the tonicity is similar to the plasma. Plasma osmolarity is around 290 milliosmol per liter. Osmolarity is calculated by this formula: 2 into sodium level in the milliequivalent per liter and 0.055 amount of the glucose and milligram per deciliter and 0.36 blood urea nitrogen level in the milligram per deciliter. We can calculate. The normal osmolarity is the 290 milliosmol per liter. The isotonic solutions, fluids having osmolarity same as the plasma, the 290 milliosmoles. Red cells are suspended in the solution, do not shrink or swell. The 0.9% sodium chloride and 5% glucose are isotonic solutions. So routinely we use in the hospital setup for as IV fluid, 0.9% sodium chloride and 5% glucose. These are the isotonic solution. So it will not affect the shape and size of the RBC. If you use the hypotonic solutions, RBC in the fluid enter into the hypotonic solution to the RBC, RBC swell and hemolysis, destruction of the RBC will occur. If you use the hypertonic solution means RBC size will decreases and water is move outside from the RBC to the hypertonic solution. So they all depend on the osmosis. Bulk flow is the along with the solutes, ions, more amount of the water, greater quantity of the water also filtered. That is called as the bulk flow. Filtration results in movement of the greater quantity of the water is called as the bulk flow. The solvent tract, here opposite. During the bulk flow of the water, it carries the dissolved particles, large amount of the solutes along with the water. That is called as the solvent tract. These are the types of passive transport. Filtration is the other type of passive transport. Here movement of water solutes from the area of the high hydrostatic pressure to the low hydrostatic pressure. This mainly occur in the kidney renal nephrons, in the glomeruli and arterial end of the capillaries. So the filtration is occur. The vesicular transport is a special type of transport for the macromolecule. It needs the energy and calcium dependent. The types of vesicular transports are exocytosis, endocytosis, pinocytosis, phagocytosis and receptor mediated endocytosis. Exocytosis is the type of vesicular transport. Here the vesicles inside the cell attach with the cell membrane and expulsion of the contents mainly occur in the neuromuscular junction. Acetylcholine is bind with the vesicle format bind with the cell membrane, releases on the synaptic cleft, the protein secretion from cell. These are the exocytosis. Endocytosis is the opposite effect, invagination of the portion of the cell membrane, trapping the macromolecules from the outside, forming the vesicle around the substance. So this is the endocytosis. It forms the vesicles. Okay. Pinocytosis, otherwise called as the cell shrinking. Okay. The soluble substances are type of endocytosis. Okay. So visible only under the electronic microscopes. The examples are immunoglobulin crosses the capillary wall. The thyroglobulin enters the follicular cells. These are the example for the pinocytosis or cell shrinking. The soluble substances are engulfed. The phagocytosis play the important role in the immunity. Here, for large insoluble particles like dead cells, bacterium, tissue debris, these are engulfed by the tissue macrophages or WBC. It is called as the cell eating or cell engulfing by absonation. Absonation means attracting that bacteria by some chemical substances, by complement and antibody. Phagocytosis means cell eating. Receptor mediated endocytosis is a special type of vesicular transport. Here, clathrin coated bits on the cell membrane. It is helpful to transport the LDL cholesterol and nerve growth factors. Okay. This is the receptor. Transcytosis, otherwise called as the cytomemsis. It involves both exo and endocytosis. Proteins crosses the capillary endothelial wall by the endocytosis and interstitial site by the exocytosis. Both endo and the exocytosis occur in the transcytosis. That is called as the cytomemsis. So transport is the active transport and passive transport. The passive transport diffusion is one type of transport. It has two types, simple and facilitated diffusion. Simple, both are passive transport. Simple diffusion for the smaller molecule, facilitated diffusion for the larger molecule. Facilitated diffusion need the carrier protein. Here no carrier protein. The Vmax is occur in the facilitated diffusion. Here depend on the concentration gradient. The simple diffusion transport the oxygen, carbon dioxide, alcohol, lipid soluble substance, and limitedly the lipid insoluble like urea, sodium, potassium through channels, protein channels. Here Vmax is present in the facilitated diffusion. Transport, the examples for the facilitated diffusions are glucose and amino acids. Differences between the passive transport and active transports. In the passive transport, no expenditure of the energy. Here uses the energy. It is a move along the concentration electrical pressure gradient. It move against the concentration gradient. Passive transport, carrier may or may not be required. Here carrier must needed. Here rate is proportional to the concentration differences. Here transport maximum, that Vmax is occur in the active transport. Classification of the transport across the cell membrane and summary. Passive transports are simple diffusion, facilitated diffusion, osmosis, filtration, bulk flow, solvent track. The active transport is again divided into the primary active transport, secondary active transport. Vesicular transport is exocytosis, endocytosis, and transport across the epithelium by it is through the transcellular transport and paracellular transport. So epithelial lining is the transport is occur through the transcellular epithelial transport or paracellular, in between the two cells the transport is occur. Thank you for watching this video. If you like this video, share your friends. Those who are not subscribed this channel, please subscribe. Thank you once again.